So let me quickly introduce our speaker today. So Daniel Margulis is a researcher at CNRS. He just told me that he has recently moved from the ICM to formerly Paris 5 at the Rue des Saints-Pères. Daniel has specialized in an approach that combines brain anatomy, and the study of brain functioning. And in particular, he has proposed these interesting ideas that brain functioning is spatially organized in the brain in the form of gradients. So he came 2 or 3 years ago at Eurospin to present his work, and I think that today he's going to present again its approach and some updates since his talk 3 or 5 years ago. So thank you very much, Daniel, for joining today, and the floor is yours.
Thank you. Thanks, Florent, and thank you for the invitation. And yes, that is exactly what I'm going to do today, which is to really give, if you were here 3 years ago, I hope this will be an interesting review of the material. And then towards the end, I'll present some of the updates and directions that I'm taking this work in the coming years. And of course, really happy for any discussion or ideas that'll emerge from this. But yeah, to sort of start things off, I guess the core challenge that I've been addressing in research over the past several years has been trying to understand the basic principles underlying the kind of complex connectivity that's just schematically represented by what you see in the background of the image here, and trying to make sense of this tangle of connections that we have in the human brain, and understanding then how that relates to the shape of the brain, to the space of the brain, and what the interaction is with cognition, with how that constrains and enables different forms of cognition. So with that as the kind of main theme, I'm going to kick off with a metaphor that I'm a big fan of. And if you've seen me speak before, you know where I'm going with this. But I think that it is a valuable kind of perspective for reflecting on where we are as a field at the moment. And if we go back about 100 years, there are all these examples across different fields, looking at the relationship of different features as they're laid out on the continents, right? So on the one hand, you have geology, in which there are these very old rock formations that are located across quite disparate portions of the globe. And on the other end of the disciplinary spectrum, there are the observation of different fossils that were found in different continents. And this was on the one hand, a challenge for all of these individual fields that were focusing on this question, but like really crucially, was the point that there was no recognition that there was potentially a common underlying answer that could account for these different features. And Alfred Wegener came along about 100 years ago and proposed a theory that we're all familiar with today of continental drift to account for this distribution of features. And while not all aspects of that theory turned out to be correct, importantly, it helped to recognize that there was a common underlying question across these fields, but also to provide a basic dynamic model that could give rise to the different patterns that were observed. And the reason why I think that's a valuable kind of historical scientific metaphor for where we are today is that we have all of these different methods at the moment for gaining access into the distribution of features across the human cerebral cortex. And these are just 3 examples among numerous that could be taken at this point that characterize in really unprecedented resolution aspects of microstructure using non-invasive techniques, connectivity in various forms, as well as distributed function. And the challenge becomes how to make sense of this similarity, how to try and integrate these different forms of data. Traditionally, over the past century, the way to do that has been through parcellation, has been through the notion that we can divide the cortex up into relatively homogeneous portions of the cortex, in which within that kind of bounded region, the features are more similar than they are with areas outside it. And that's exemplified in many ways by the recent landmark work of the Human Connectome Project, Mac Lasser and co-authors publication of a new cortical parcellation that's really established a new foundation let's say, or a new step in the field. It's the culmination of many years and many decades of research in a broader sense. So what this allows us, what this kind of availability of these different modalities, the accessibility to different both approaches, but also just the wealth of data, is we can begin to ask a different kind of question. Rather than purely asking the question of what are the boundaries of different regions, where are they constrained, we can begin to look at questions of why are they spatially distributed as they are. And this actually goes back to a question that was brought up by researcher Friedrich Sinides around 60 years ago now, who asked the basic question of what are the spatial trends that we observe in different cytoarchitectonic features as they're distributed across the cortex. And Sinides was trained in basic techniques for microstructure, for parcellating the cortex using microstructure. But rather than continuing to focus purely on where the boundaries were, began to ask this overarching question. And it's again that shift, that kind of Wegener-like shift from how do we describe boundaries to how do we talk about spatial relationships? What is the origin of those, and what is the consequence of those? That I think is a really interesting question for us to begin to also ask within the current moment in human brain mapping. So I'll give a very quick overview of Sinides' ideas, not because I'm going to be focusing on it so much in the talk, is that I think it's just an interesting way of approaching the problem. The basic idea that he proposed is that the cortex emerges from two trends of progressive laminar differentiation, meaning that the cortical layer structure becomes increasingly more complex as you move away from these two starting points, one being hippocampus, that you can see here on the slide, the other being the piriform cortex or olfactory cortex. So essentially there's these two different halves of the cortex, one being this kind of memory oriented conceptual, let's say, portion, the other being a sensory related function. They're very early types of cortex, 3-layer structures. It becomes increasingly more complex as you move spatially further away in these concentric rings that you can see in the images on the screen here. And that they meet essentially in the middle. You have a point where the two kind of waves of progressive laminar differentiation then converge along the lateral wall. And this line of research, this way of thinking, was actually adopted through interesting kind of historical accidents. Sinides spent a fellowship at I believe Mass General in the States in the lab of Deepak Pandya for a year, and was able to transfer these ideas to the field of macaque-monkey track tracing connectivity research, which is our gold standard for characterizing connectivity in the primate brain. And one of the key ideas that came out of that interaction and followed up by members of the Pandya lab, Helen Barbas and others, is that areas that demonstrate similar degrees of cytoarchitectonic differentiation, so again, similar levels of complexity relatively, show preferential connectivity between each other. So this is just, again, a schematic to illustrate this point, but as you move along the frontal lobe onto the lateral wall, areas that are showing similar types of patterns within the microstructure are more likely to connect. And the kind of pithy summary of this principle in the field is like connects to like. So what this did was to connect up the microstructure notion with connectivity. And it's bringing these two modalities then together to look at how they're converging and what principles might be underlying that relationship, that similar spatial relationship that's observed. Another person to pass through this lab and be exposed to these ideas was Marcel Messelem, who in a series of papers really synthesized this line of work across multiple scales. And one of his landmark pieces in Brain, 1998, brings it together beautifully. Also reflecting on the clinical implications of these organizing patterns. And one of the key ideas that he proposes is this global hierarchy in which we have a segregation across different sensory modalities and integration taking place hierarchically towards a core set of regions here represented by the connections in red that represent the integration across these different modalities, a kind of higher order representation. Crucially, what Mesalam did here was to take what was known about primate connectivity from the track tracing literature and to hypothesize what would be the case in the human brain based on what was known in the macaque. But this was not based on human data. It was purely a theoretical kind of schematic based on what is known from other species. And so where I'll be going with the talk today, and sort of bringing it back around to is where we are really in the field. And I come from cognitive neuroscience predominantly, and I think one of the key challenges is that we're in many ways used to thinking over the past few years in terms of networks, in terms of large-scale homogeneous systems, a kind of distributed version of the module notion in cognitive neuroscience. And one of the key challenges that has interested me is this: how can we think about a model of large-scale connectivity and cortical organization that incorporates both the global networks that we're used to thinking about with these notions of local processing streams over the cortical surface? And so that's one of the themes that I'll also be addressing. So in general, though, just to kick things off, what are the spatial principles that underlie these different features and how they spatially converge? So far, so good. Are there questions? I mean, feel free to jump in. Just Flora, if you don't mind, just keep an eye if anybody has anything, any comments come up. Great. So to kick things off, I'm sure everyone's familiar. This is resting state fMRI as far as the paradigm is concerned. So it's pretty easy to go through the paradigm side of things. And this is where I got my start in mapping, in human brain mapping as a field. What you see here is a video that's been created of activity during a resting state scan. And it's really just the raw data. It's percent fold signal change. So you're seeing reds being higher levels of activity, blues being lower, and green somewhere in the middle. And it's these very slow fluctuations. This is essentially real time. It's been smoothed over time, but these very slow fluctuations are actually what we're acquiring when we're acquiring resting state data. And the challenge becomes to make sense of the spatiotemporal patterns that we're observing in the data during this state. And these patterns are ubiquitous, of course. They're not unique to resting state, but I'm sure I don't need to explain that to this audience. So one of the core agenda within the field has been using this for whole brain parcellation, for being able to divide up different systems in the cortex based on this data. And for those unfamiliar with these maps, you have to trust me on this, but there is a remarkable amount of spatial consistency across these different patterns. And it's regardless of where the site that the data is acquired at, the individuals, even to a large degree, the techniques that are used for dividing up the data set, for clustering it into these different patterns, for distinguishing different networks. There's a high degree of spatial consistency in the basic pattern of networks that are observed. One of the key differences that's found across these approaches is, let's say, a consistency in the number of networks that are extracted. And in some ways, I think that's one of the themes that'll come up later of what are some other approaches that allow us to move beyond the question of what are the specific boundaries of these networks, to asking the question of what are the underlying patterns that we're dividing up in different ways, and how can we characterize that in a meaningful way, and what's a language we can use within cognitive neuroscience to talk about it, which is one of the challenges, of course. In my own work, I've focused in rather than on the whole brain connectivity question, more focusing in on looking at subdivisions within heterogeneous portions of the cortex using functional connectivity. So early on, we did work on anterior cingulate, finding subdivisions as you move rostral caudally that were consistent with the meta-analytic literature. And then moving on to doing some cross-species comparative work, looking at the posterior medial cortex, and finding consistencies with what was known in the track tracing literature in macaque monkeys. And then, I guess not so recently anymore, but more recently in the other two, looking at ventrolateral prefrontal cortex. or ventrolateral frontal cortex, and being able to distinguish basic patterns as we move along that within on the individual level also. And also developing techniques for mapping high resolution for manually delineating regions using high resolution visualization techniques that are consistent with the methodologies of the traditional neuroanatomy, but trying to bring them to the kind of data that we're used to working with today. So I'll focus in on the ventrolateral prefrontal example because it helps to set up the techniques that I'm going to be focusing on for the gradient themed work in a moment. Just very briefly, connectivity within ventrolateral frontal cortex. This is the macaque monkey cortex. It's a schematic where you see areas represented by these different colors on the lateral surface. And we can represent the connectivity between these regions, again, schematically based on literature. What occurs is that as you move further and further away from the central sulcus, areas are preferentially interconnected with one another. And this is just a general schematic. So all areas are connected with their neighbors. And I will get back to that point later on. But all areas are connected to their neighbors. And then when we're looking at the long distance connections, there seems to be this basic kind of rainbow shape in which as you get further away from the central sulcus, areas are more likely to be projecting one another. And that's represented here. And we can use that information then to distinguish different portions of the ventral lateral frontal cortex from one another. So this is a basic kind of model of how that connectivity would be arranged, CS being the central sulcus. And we can take seed regions now, so regions of interest within this portion of the cortex in the human brain using morphological landmarks. And this allows us then to distinguish these different areas from one another. What you're seeing here is the average results across 6 individuals. And what was exciting about these results in part is that it very clearly mapped onto what has been observed in the macaque monkey trap tracing literature. So we see this basic progression from area, here's the part with the, hold on. I'm not going to be able to point, it seems, but where we're able to move from area 4 being the yellow one to 6VR to 44, 45. And we see that same basic pattern of yellow to blue to green to red occurring in the inferior parietal lobe. And so that was consistent. There's extensive connectivity of area 45 within the superior temporal sulcus. What we didn't really expect and what was kind of interesting and provocative for us to see was that same basic pattern of yellow to blue to green to red was repeating throughout the cortex. And it raised a question of what are potential ways to identify that repeating pattern that don't rely on a priori hypotheses regarding the starting points, the seed regions, that don't make any assumptions about the boundaries, but simply characterize that spatial trend that we're observing across these different networks as we step across them. So to kind of present this in a different way, the traditional approach to characterizing these kind of network subdivisions would be to represent the brain as a graph in which each of these black dots represents a point on the cortex. This is a schematic, but each of the black dots represent a point on the cortex, and their spatial proximity represents how connected they are, let's say. And the traditional way to approach trying to understand the layout of connectivity patterns would be to subdivide this into portions that are more interconnected within those areas than they are to ones outside, to essentially cluster it into different networks. And instead here, the question that we had was, how can we characterize the basic patterns across this graph? And instead we can use what comes down really to a fancy version of principal component analysis. It's a nonlinear dimensionality reduction technique called diffusion map embedding, but you can really think of it as doing a principal component analysis to pull out the dimensions that represent the maximum variance in connectivity patterns and project all the data onto these different dimensions. So here's just showing the first 2 again schematically, but it becomes a way to try and capture as much information as we can in a low dimensional space. And for folks who are interested, these are the equations that represent diffusion map embedding. It's very similar to Laplacian eigenmaps. What we have found, I think it's really crucial to see, collaborator Sukjun Hong has recently published a paper showing that, and I think it's very exciting to see, that essentially principal component analysis gives you very similar results in this case. And this is a great, actually, this is a good time to say that I've had some conversations with folks at Parietal years ago that suggested that this might be the case, and it was correct. So thank you for that. And what's good in a sense is that I think there was a bit of a black box surrounding the method. So the crucial point is that it's nothing specific to the method that's deriving the patterns you're about to see in a bit. It's really just present in a low dimensional embedding of the data using linear techniques as well. But again, just to illustrate for folks who aren't familiar, the idea is that we're able to reorder the connectivity matrix in a way that places patterns that are more similar to each other at the proper ordering along that matrix. Okay, so now if we apply this to the human connectome data, I spin those over there. What we see is that it represents, now this is taking the first two dimensions representing the highest variance in the connectivity matrix using functional connectivity from the human connectome project. And what we observe is a pattern which there's differentiation, similar to what Messelum described, differentiation along the second gradient between sensory modalities, and then integration towards these regions in red that we can consider as a kind of association cortex or transmodal cortex. What I'm going to do now is zoom in specifically on that first gradient, or what I'll term the principal gradient here. And what we see is that at the peak of that triangle, at the peak of that kind of integration across different modalities, are these regions in red that are characterized in the human as the default mode network. So what we appear to be observing is this basic pattern where at one end in blue we have sensory and motor networks regions, and at the opposite end of that spectrum are areas of the default mode network. And in fact, if you take a kind of canonical set of networks, this just being one representation from the Yeo-Creenan 7 network parcellation, and you take the values from the principal gradient and simply represent them in this blocks plot, what you can see is at the far left, the purple and blue, the visual and somatosensory motor network, followed by a kind of attention systems, the frontal parietal in orange, and finally the default mode in red at the opposite end. And this is also consistent with what would be observed spatially. So essentially, if you were to drop down on any point in the cortex and look to your left and your right, you would see the same, your spatial position within this kind of hierarchy would allow you to predict who your neighbors are at any point in the cortex. If you fall onto the orange portion or the orange network anywhere in the cortex, you're going to be flanked by red on one side and green or pink on the other. And that kind of relationship back to space is something that I think is one of the key themes of this relationship between large-scale connectivity and cortical geometry that we'll get back to as well. So it allows us to represent and to think about these networks rather than being kind of distinct, completely segregated systems, as thinking about their relationship across one another, and being able then to situate them in this sort of hierarchical model similar to what Messelum has proposed. And one of the key points of this line of work, I think, that in some ways helps to kind of extend or update the Messelum model is the representation of the default mode network at the center of that space. Because the default mode network in 1998 wasn't even a twinkle in Marcus Raichel's eye. It was really an emerging concept. And rather than it being this kind of structure that's completely independent of any other functional system, it allows us to begin to think of it as one endpoint along the spectrum. And it raises, of course, challenges with that, but at least provides a context that that we can use to understand what its role might be. So I'll go through the next portion quickly, just in the interest of time. And the next portion of the talk, I'd like to just mention a few lines of work that are addressing the question of how does this converge now with cortical microstructure? How does this relate to the basic kind of features that we can describe using MRI of the cortical layer structure? And as I mentioned at the beginning, there is this observation that goes back a few decades of like connects to like. So areas that are more similar cytoarchitectonically are preferentially interconnected. We wanted to explore that using human data. It's been observed across multiple species where the data is available, but in humans it hadn't really been directly addressed. And one of the ways that we can do that is to characterize human cortical microstructure using T1 maps, which act as a proxy for intracortical myelin density. And of course, it's a controversial topic, but at least it provides us some quantitative measure for characterizing features of microstructure in the human. And this is just some evidence. So intracortical myelin has been used in a meaningful way for representing different portions of the cortex, as shown in the left, and this goes back to Flessig's work from the 1800s. The technique itself has been validated. This is in Leipzig, where it was based before, and can be investigated non-invasively using MRI. And again, also recently, another approach using the T1, T2 weighted ratio by the folks at WashU has also been shown to be relevant for characterizing subdivisions in the cortex, so using it for parcellation. Here we want to do is acquire high resolution data, and this is work done by Julia Hudenberg, who was a doctoral student in my lab. And what you see on the top is across 8 subjects, the T1 maps from those individuals. And here's the functional connectivity from that same data set. And what it very briefly she observed was this relationship between the T1 values and the principal gradient in that data set. Oh, sorry, that was fast. Importantly, not all aspects of these mappings converge. For instance, there's difference within the lateral parietal lobe that was not accounted here in the data, but it provided a kind of starting point to be able to use further techniques to, in a more detailed way, characterize the relationship between histology and connectivity in the human brain. And Casey Pakwala has been doing absolutely beautiful work with Boris Bernhardt's lab in Montreal, looking at a technique. This is just one of the techniques they've introduced, but essentially looking at the big brain data set, which is this very high resolution histology data set of the human brain, and being able to relate that then to MRI-based measures. And they've developed a technique called microstructure profile covariance, which is looking across the cortical layer structure at different features and characterizing these profiles and how they relate to one another across the whole cortical surface, which then provide a basis for describing similarity and gradients in the similarity patterns. So what you see on the left are is the principal gradient based on histology values from the big brain data set. And here in blue is the relationship of that to the MRI-based measures, and essentially provides a basis for, again, describing with more detail the specific relationship between, in this case, MRI and histology, but focusing in on variance in microstructural features. So it appears that there is this consistency in a large-scale pattern that's being observed in connectivity, in microstructure. And the next kind of set of questions that I'll go into is, what are the basic, how does this emerge? Why is it that we see these spatial convergence, this similarity in the layout of these different features? And I'm a big fan of this paper by Buckner and Kreenan. It was really see, it elicited a lot of ideas in the lab when we first read it back in 2013. And Buckner and Crean were interested in a question of why is it that we see such dramatic expansion of association cortex in the human brain? What can account for that? And what they propose is a theory called the tethering hypothesis. And I'll explain that very briefly in this slide here. The notion being that in the ancient mammalian cortex, very small cortex, there are, during development of the cortex, there are molecular gradients that determine the specialization of different regions. There's a reinforced bythalamic input, and those areas differentiate to become specialized for the specific sensory modality that they're involved with. But then we have the dramatic cortical expansion that occurs across evolution. And by the time you arrive at the human cerebral cortex, there's still the same molecular gradients that determine the specialization of different areas. There's still the same thalamic inputs that really render those concrete. However, now there's all this additional cortex that's in place. And it is not constrained by those features that determine the specialization of primary cortex. It becomes untethered from them. And instead, it is the connectivity between those regions of cortex that gives rise to their specific functional attributes. Right, in a nutshell, tethering hypothesis. But what this suggested when we read it, and was kind of exciting for us, is that implicit in the hypothesis is the idea that that the space of the brain would play a crucial role in how these different cortical types were spatially arranged. So if it's correct, one could assume that there should be a very precise spatial mapping between the aspects or features of association cortex and location of primary regions. And so that's what I'm going to show here in a analysis where we simply look at the distance along the cortex from the peaks of the principal gradient. So the white dots that you see here are the local maxima. We're just now measuring with like a tape measure along the cortical surface. And what you're going to see in the next image is purely based on that geometry. It's just a measure of the cortical geometry. Here's the basic map. It's geodesic distance along the cortical surface. The gray lines represent the equidistant positions between those dots. And what you can see is a really, really neat kind of spatial convergence with the morphological landmarks of primary areas. So CS, the central sulcus there, these areas along the points of the default mode network appear to be equidistant from the central sulcus and oriented in a way that that equidistant line really converges with the orientation of the central sulcus as well. Same with the calcarine sulcus, or the landmark of primary visual cortex. Right? And to a lesser degree, but to some extent as well, the transverse sulcus representing the position of primary auditory. And so it appears that there is a really precise spatial relationship between the points of the default mode network, such that they're in some ways as far as you can get from the location of primary areas. The system that's involved in let's say in memory related, abstract, internally related functions is as far as possible from a system that's essentially determined by interaction with the environment. And we'll get back to that in a moment. But like one of the aspects of this that I find interesting and exciting, and is leading to the current work that I'm taking forward, is that in some ways this suggests that the local organization of the cortex provides an organizing mechanism. And one of the, I think, what does it mean that we have this relationship with the local geometry and the distribution of large scale systems? I think one aspect is that we can think of local geometry as representing local connectivity. And I'm actually going to skip a couple of slides. No, I have it later, sorry. But as representing local connectivity. I will get back to that point. And that the other aspect of this, I think, is crucial, is that we're used to dividing the cortex up into different areas, thinking of those as having a specific functional role, and then let's say thinking about the hierarchy of those areas as a whole. But what this potentially suggests is that there might be a mechanism as well for local interaction, irrespective of different areal boundaries. That we don't need to divide the cortex up into distinct computational units to be able to reflect on what the computations that could be performed over the surface might be. Yeah. So cool. Yeah. So stop there. The is this conserved across phylogeny? I see I'm watching the clock, and so I'm going to continue to move. But again, if there are questions, I'm happy to break into discussion whenever folks want to do that. Is it conserved across phylogeny? I'll fly through these slides because I've already, I think, touched on it a little bit. But we've also looked at this across different species, and this has been kind of a long-standing interest. It's become more and more accessible with the availability of data through consortia such as PRIME-DE. But one of the key points is that we were able to observe this using track tracing data in the macaque monkey, which you see on the right, using the CoreNets database made available by Henry Kennedy's lab. And we see the same basic observations of convergence with the T1-T2 ratio in this case, so our proxy for intracortical myelin. And really crucially, and this I find quite exciting, is we see that same basic relationship with the distance. So you look at the distance from the local maxima of the principal gradient, and it converges again with these landmarks of sensory motor cortex, which I think is quite remarkable that we're seeing that in another species so beautifully here. And again, is provocative. Why is it happening? No. So this is one of the areas that I'm quite excited about following up on, the degree to which this is a consistent feature in primate cortical organization. If it doesn't extend beyond primates, why is that? How does this emerge, etc.? Yeah, please.
Daniel, can I ask a question?
Yeah,
you're always showing a left hemisphere, and I wonder what happens to the right hemisphere, and especially whether there are asymmetries in the human that you would.
There are. Yeah, that's a great question. Yeah, there are asymmetries, and the reason I show a left is because there's only so much space on the. It's purely to make it easier to follow the story. There's only so much space on the screen, and there are asymmetries. There are more consistencies than asymmetries, as always with this. But even if you go back to the Yo networks, none of this is new in the sense that it's always been there in the data, these observations of this kind of ordering of patterns. And you go back to the famous Yo-Creenan networks that we're used to looking at, and you can already there see the asymmetries between the right and left hemisphere that become present in, or that are also represented in this kind of connectivity gradient space, whatever. The other key thing though to mention is that we've been looking recently at differences in the geometry, and you do see interesting differences in the distance of the, for instance, the angular gyrus to primary sensory regions between right and left hemisphere, and it provides also a good starting point for controlling across hemispheres. Of course, you can look at the differences in hemispheric geometry on the individual level. So we're doing that as well. But yeah, Beth Jeffries has been looking at those asymmetry relationships and finding some interesting stuff.
I suppose you don't see any of these asymmetries in the monkey?
It's a question that's come up. I don't know. There is handedness, right? So there may be certain asymmetries, one would imagine to a lesser degree, but yeah, any thoughts on that? Or
I think what you would expect is there may be individual asymmetries, but there would not be a systematic asymmetry.
Right. Okay.
If I may ask a question as well, because
please, yeah.
Oh, yeah, well, I love it. I mean, this idea of geodesics. I don't know if you're aware, I published a paper last year on a new model based on the relativity theory. where I also introduced geodesics. I think the idea is very similar to what you propose, except that I introduce a time. So not only I consider that there is a functional distance between regions, but it depends also on how fast information is exchanged within the networks. So I propose what I call a brain space-time concept. You might be interested to. I will send
super interested. That'd be great. Yeah, that'd be fun to chat about that. Yeah. Thank you. Cool. Great. Are there other questions at this point? I always think it's fun to get the discussion going. I've got more slides I can continue to go through, but if folks do have questions, jump on in, and I'll keep going. Florent, just give me a nod if I should. I'll keep going, and yep, great. Thank you. Cool. So quickly again, we've also taken this to the marmoset monkey, and for folks who don't know this data set, it's really a remarkable resource that's been made available by Marcela Rosa and Piotr Majka. And very briefly, we took this data set and began with a kind of qualitative comparison of the different track tracing injections in the marmoset monkey and observed a similar pattern that we've observed in the macaque and human, represented on the flat map of the marmoset on the right here. And this is a more, let's say, a data-driven approach to that same observation. But very briefly, as you've guessed at this point, the theme, it's the same song, slightly different tune. We observed the same basic differentiation of sensor modalities and convergence towards transmodal regions in the marmoset. One of the interesting observations here was a difference in that basic pattern that was observed in the temporal lobe, which does raise certain questions about what's occurring there. There are some unique features of temporal lobe organization in the primate brain, geometrically as well, and it raises some interesting kind of, let's say, hypotheses for future inquiry there. So we have these different models of, or let's say, results in different species, which allow us to begin comparison across different species and how cortical expansion would occur within this space. There's been a lot of exciting recent work looking at cross-species comparison. Ting Hu has been doing beautiful work comparing humans and macaques. Rohir Mars as well, also looking at chimpanzees, if I recall correctly. And it raises the possibility for thinking about the question of cross-species comparison, cortical expansion, using additional modalities to characterize these different spaces. So I think I'll really fly through this because the basic take-home message I've already said in different ways, and I'm sure folks are already kind of getting the main points. I won't hit it too many times over and over, but we've also taken meta-analytic approaches, and as you guessed, even with the meta-analysis, you get the same idea of sensory motor on one side of the spectrum, domain general in the middle, and kind of memory-oriented conceptual higher order processing at the opposite end of that spectrum. And the one point that I'd like to mention here that I think is fun is if you think back to that Sinides idea from the beginning of piriform and hippocampal cortex being opposite ends of the spectrum. Thematically speaking, it's similar to what we're observing here, in which we've got memory-driven higher order default mode at one end of that spectrum, obviously more complex than purely hippocampal, but potentially incorporating it. And at the opposite end, sensory input and motor output. So that same basic spectrum being captured here by connectivity and consistent with the functional literature. We've taken various approaches, and I'll just flip through this, but we've taken various approaches to using this as a way of representing the complex spaces that we observe during different task states. In this case, looking at stimulus dependent, so decisions based on content that's visually available in this case, that's visually available versus making a decision based on content that's only accessible in working memory. And looking at the difference between those states and how those states can be represented, or how the state changes are represented as a shift along the principal gradient. And so by comparing and contrasting those states, we can see that on the right, there is it's distinguishable by the position along this kind of sensory to memory spectrum. I'm sorry, I'm going through this quickly because I'd like to get to the last part. So if you'll bear with me, I'll just jump ahead rather than talk through this too quickly. Summary slide, network organization emerges. In a sense, there's this tight relationship, not from maybe, but there's a tight relationship with local cortical structure. We can begin to think about these connectivity patterns as recruitment along this gradient. And where all of this kind of is going in my work and what I'm thinking about at the moment is, how does this relate? The fact is that like connectivity is predominantly local, and this goes back to work from Kennedy's lab and others. We really like to focus on the long distance connections, you know, in my work, absolutely as well, because they're interesting. Because it's when you have a network structure, it's interesting that things are talking to each other that are very far away from each other. So, it requires enormous energy expenditure. It's unique. It allows us to characterize the differences. But the fact is, the vast majority of cortex is talking to its neighbors, and that is captured by local geometry. And so, how can we return to that idea of what might be like the rather unique features, looking at what is the ubiquitous features and trying to understand what the role of this local geometry might be, is kind of what its impact might be on cortical organization cognition. I'm really fond of these images. Again, just in the interest of time, I won't go into this in detail, except to say that this is a really beautiful concept from Humboldt from 150 years ago, which he represented different species of plants within the terrain of these mountain landscapes. And what was interesting about it as a kind of academic exercise is the notion of representing speciation or distribution across space. And asking the question of what is the interaction between the environment, latitude, altitude, and biological species. And essentially, I think, again, in a very broad sense, we're primed to be able to ask a similar kind of question about what is the relationship between various features of space, and how can space and the distribution of those features help inform us about the underlying principles and mechanisms. So what we've proposed in general is that some of these ideas might begin to constitute a kind of coordinate space. Rather than arbitrarily dividing the brain up into X, Y, and Z coordinates, we can begin to talk about positions along the cortex as having meaningful functions, or providing a context for a functional interpretation of positions along the cortex. And so here, principal gradient in the center, that I've already discussed extensively. And then on the right, this is the notion of relative distance from different sensory modalities, different combinations of sensory modalities. And that's really the core of the next kind of wave of research that I'm hoping to carry forward or begin working on, which is this idea of, you know, the principal gradient. It's just one dimension. It's been really interesting to see the ways in which it converges, but it's just one dimension of representation. And obviously the brain is far more than that. So one of the questions that's come up for me is that within the principal gradient idea, within that one dimension, there's nothing that allows us to differentiate there's nothing that allows us to differentiate transmodal regions from one another. What I mean by that is that those regions in red that you see, they all converge towards the peak of that triangle. And from that space perspective, they're equivalent to one another. But they're not doing exactly the same thing, as we know from the cognitive literature. And so one of the questions I want to get into is what might account for the differentiations observed across these portions of transmodal cortex. And the hypothesis is that we can begin to potentially think about them as being distinct convergence zones of different sensory motor combinations of input. And funny enough, this goes back to an idea from Damasio in a sort of more abstract way from a few decades ago, which he proposes a theoretical approach to addressing the binding problem of convergence zones on a circuit level. so that there's a hierarchical organization of the integration across different sensor modalities, and that hierarchical organization allows for back projections to coordinate sensory activity. And what it seems to me is that the macro scale architecture that we're observing with these distance measures might provide a kind of large scale structure to support that notion that the space could act as a coordinating principle if you think about activity as occurring locally along that space. So again, just a kind of schematic representing that. And the idea being that if you take into account the relationship with these distances between the peaks of transmodal cortex and primary areas, one can begin to think about the locations of transmodal cortex, the speaker, is representing the distinct combinations of different sensory inputs as they converge over the cortical surface. And that's schematically represented on the left. So in the last couple of minutes, and I'll try and leave time for questions, I'll just show some pretty pictures. So these are inflating the brain now. These are arrows that are capturing the same data, but are representing visually now the kind of spatial convergence towards the peak of the principal gradient here. You can see all the arrows pointing in towards the peak regions. And we can use that basic data set to conduct, in this case, a surface-based watershed segmentation, which represents the basins over the cortical surface of convergence. And I've termed these integration zones, and then found out about Damasio's idea of convergence zones, which seems like a much nicer term to use. And this is the basis for this hypothesis that I'm playing with at the moment of how can we begin to think about these zones as representing the convergence across different combinations of sense modalities, and to what degree can that account for the distribution of functions that we observe over the cortical surface. So again, I'll just kind of go through this quickly, as it is a hypothesis at this stage, but the basic idea being that if we represent different portions of the cortex along these two basic dimensions, one being the principal gradient, the other being the relative position between different sensory motor areas, it potentially provides us a framework for characterizing the distribution of different functions within cortical regions. So to kind of schematically, again, represent this, actually this is using data, just to mention, but the temporal lobe, we can project that into the space using these two basic coordinate values. And it begins to provide a basis for thinking about the observation in the cognitive literature of semantic, in very broad strokes, semantic cognition occurring within the temporal lobe versus the lateral parietal cortex involving representation of the body, somatosensory cortex. When we think about the convergence of higher order body representation with higher order visual representation, it becomes intuitive that we see aspects of spatial cognition occurring, that the higher order kind of combination across those modalities results in abstractions such as spatial cognition, numerical cognition, navigation, etc. And so, again, this begins to provide a kind of way of thinking about the role that the space of the cortex may have in determining the distribution of higher functions, may, as I said at the beginning, constrain and enable the distinct abilities that emerge across the cortex as well. And I did want to mention, as you might imagine from the observations I showed briefly in the macaque monkey, we're observing a similar pattern there as well. And so lots of exciting things going on. I'll kind of wrap it up there, and I think I managed to stay in time. Sorry that was a bit fast at the end, but really happy to have discussions. I'm right around the corner when we're all allowed to see each other again, and super happy to continue the discussion. Just as wrap-up points, I think where all this is going is trying to think about a model-based approach for talking about functional organization. And part of that's based on this observation of convergence with connectivity and cortical geometry. I do think a key point that is of value is being able to redefine the default network, which has become and been quite a challenging term within the field, as being the one end of this hierarchy of organization. It allows us to compare across species and to describe it independent of the numerous different ways that it's been defined, and that make it controversial in cross-species comparative work, especially. And I think bringing it together, thinking about a neuroanatomical basis for talking about functional specialization. So cognitive neuroanatomy, essentially, where you can talk about building blocks of different functions as emerging from large-scale features of cortical structure. And with that, I'd like to thank my former lab members in Leipzig, wonderful collaborators. All the data and that's ours, at least, and the code is available at the link on the bottom. It wouldn't have been possible without the data resources. And thank you. Thanks for joining. And like I said, happy to discuss anything, and feel free to get in touch.